Okay, so now we're going to talk about metabolism and nutrition. Um, Energy content in the human diet is measured in terms of heat, and a calorie is measure of heat. 1 calorie is the amount of heat required to raise the temperature of 1 gram of water by 1 degree Celsius. A kilocalorie is an actual unit of calorie that's used to measure human diet. 1 kilocalorie is the amount of heat required to raise the temperature of 1 kilogram of water by 1 degree Celsius. So metabolism is the sum of all the body's chemical reactions. It's a series of enzyme catalyzed reactions and metabolic pathways, which include harnessing energy in chemical bonds of molecules obtained from diet, which is our nutrients, that may be used to make ATP, converting one type of molecule into another for cells synthesis reactions, synthesizing macromolecules such as proteins, polysaccharides, nucleic acids, and lipids, and breaking down various macromolecules into monomers and other smaller molecules. Now there's two phases of metabolism. We have catabolism and anabolism. Catabolism is a series of reactions in which one substance is broken down into smaller parts. This usually involves breaking bonds, in which case you're going to end up releasing energy in the form of ATP. Glucose is broken down it's the breakdown of dietary and stored carbohydrates. It's carried out by every cell in the body. Glucose is a preferred fuel for many cells, including that of the brain and the liver. Fatty acids, which are typically stored as triglycerides, are broken down into free fatty acids and glycerol. They can proceed into separate catabolic pathways or can be used by the cell for other purposes. Amino acids, these get liberated by catabolic pathways that break down proteins. However, instead of the amino acids being used for energy production, they actually get used by the cell to make new proteins, usually, unless you're under conditions of severe starvation. Anabolism, on the other hand, is the second phase of metabolism in which you have a series of reactions in which smaller molecules are combined to make larger molecules. Cells use smaller molecules to build larger macromolecules, such as proteins, nucleic acids, lipids, and carbohydrates. Chemical reactions are usually paired such that energy is released from an exergonic reaction, usually from a catabolic process, that feeds liberated energy into an endergonic reaction using an anabolic process. Now, the law of conservation of energy states that energy cannot be created nor destroyed. It can only change forms. Energy present before the reaction in the reactants must equal the energy present after the reaction, meaning both sides of the equation have to equal out here. So we have our exergonic reactions where we release energy, leaving products with less energy than the original reactants that were processed. Mostly catabolic reactions are exergonic reactions where you are releasing energy. Endergonic reactions require the input of energy to proceed. These products possess more energy than the reactants. Energy must be added to the reactant side of the equation to satisfy the law of conservation of energy. Most anabolic reactions are referred to as endergonic. So here we have energy is released from an exergonic catabolic reaction. Okay, and on the bottom we have energy is from ATP breakdown and fuels, and it's going to fuel an endergonic anabolic reaction. And in the middle, you have the production and storage in the form of ATP. So following a series of events describe the relationship between exergonic and endergonic reactions within a cell, energy is released from exergonic catabolic reactions. It's used to fuel endergonic anabolic reactions of ATP synthesis. ATP then gets broken down into exergonic catabolic reaction in which the energy from ATP breakdown now fuels another endergonic anabolic reaction. So essentially what this is saying is that you break down molecules to release energy, store it as ATP, that ATP gets used, And the energy from the ATP then fuels an anabolic reaction, meaning you're not always going from a catabolic reaction to an anabolic reaction. ATP is the middleman, and that's how you can actually store energy. And again, ATP is going to be the main form of energy currency that is used by cells. ATP is an inherently unstable molecule. Negatively charged phosphate groups repel one another strongly. This places strain on the molecule, making bonds highly unstable and a very reactive molecule. The bond between the second and third phosphate group is extremely unstable, and it releases a great deal of energy when it's broken. The bond between the second and third phosphate group also has a misnamed high energy. This possesses no more energy than any other bonds. Cells harness ATP energy by removing the third phosphate group in a hydrolysis reaction. The bond between the second and third phosphate group is broken with a water molecule, releasing the free phosphate and ADP. ATP hydrolysis is a highly exergonic, but cell is only able to harness about 40% of the release energy to perform the work. 60% of that release energy is lost due to heat in the inefficiency of this exergonic reaction. So the harnessed energy from ATP drives cellular processes in two ways. The released energy can be used by the cell directly to fuel certain reactions, such as muscle contractions, as we've talked about. And also the released energy can be used by the cell process called phosphorylation. Now, phosphorylation is a process in which ATP donates phosphate group to a reactant under the direction of an enzyme. The reactants become more reactive, and it favors the conversion of the reactant to the desired product. So essentially, you can put a phosphate on a molecule, and that could actually activate something. It could deactivate something. It could change the structure of the molecule, which could, cause it to do various functions within a cell. Now, the capability to use chemical energy and nutrients to drive ATP synthesis is the result of the ability to transfer electrons from one reactant to another. And this is going to involve electron carriers, and there's two primary electron carriers that we'll talk about, and those are NADH and FADH. And the transfer of these electrons to these electron carriers usually follows characteristics defined by oxidation and reduction reactions. When fuel is burned, its electrons are transferred from one molecule to another. This is an exergonic reaction that releases energy and heat. Substances that donate or lose their electrons are said to be oxidized, versus substances that gain those electrons are reduced. Okay? You cannot release electrons without having something else pick them up. All right? The conservation law in thermodynamics here, that's what that states, is when you release electrons, something has to pick them up. So the release is the oxidation reaction, the gaining is the reduction reaction. These two reactions always go hand in hand, and they have been dubbed redox reactions. During catabolism, nutrient molecules are oxidized and their electrons are transferred to another molecule, which is then reduced. The flow of electrons from one molecule to another is then used to perform cellular work, including that of ATP synthase. So here we have NAD, and then you have the release of electrons, which gets picked up, and you form NADH, which is the reduced form. These electron carriers are molecules with high electron affinity. They accept electrons that are removed from the oxidation of nutrient molecules. Many of these carriers are peripheral and integral proteins found in the mitochondrial membranes. And again, NAD and FADH are your two primary electron carriers that we typically see. And their job is to actually just shuttle those electrons from a pathway, such as, say, glycolysis, and they will pick up those electrons that got released by the oxidation reactions. Then themselves becoming reduced now, and then what happens is that reduced electron carrier then can travel to or shuttle to the electron transport chain, where it can then release those electrons into oxidative phosphorylation to produce ATP. That once that happens, that electron carrier is now free to go back and pick up more electrons. So essentially, these electron carriers are just shuttling these electrons between different oxidation and reduction reactions. Okay, so that's a little bit of an intro, and that'll be the end of the first recording for metabolism.